Have you ever wondered what makes you, you? Why you have your mother's eyes or your father's smile? The answer lies in a tiny, incredible molecule inside every single one of your cells. It's called DNA, and it's basically the instruction manual for life itself. Today, we're going to unravel the secrets of this amazing molecule, so stick around. So, what exactly is DNA? The full name is deoxyribonucleic acid, which is a bit of a mouthful, so we'll just stick with DNA. Imagine a massive library, but instead of books, it contains all the information needed to build and run an entire living thing, whether it's a giant blue whale, a tiny ant, or you. This isn't just a metaphor. DNA literally carries the genetic instructions for every known living organism. It dictates everything from the color of a flower's petals to the intricate workings of the human brain. It's the ultimate blueprint, passed down through generations, connecting us all to the very first life on Earth. Let's zoom in and look at its structure. You've probably seen the famous image, the double helix. It looks like a twisted ladder, and that's a perfect way to think about it. The two long, spiraling sides of this ladder are called the backbone. They're made of alternating sugar and phosphate molecules. They provide the support and shape for the entire structure. But the real magic happens on the rungs of the ladder. These rungs are made of four chemical bases: adenine, thymine, guanine, and cytosine. We just call them a T, G, and C for short. Now, these bases don't just pair up randomly. There's a very specific rule: A always pairs with T, and G always pairs with C. Think of them as puzzle pieces that only fit with their specific partner. So, if you have an A on one side of the ladder, you'll always find a T on the other. If you have a G, its partner will be a C. These pairs, a T and GC, form the steps of our twisted ladder, and it's the sequence of these bases that holds all the genetic information. So, how does a simple sequence of 4 letters contain the instructions for something as complex as a human being? Well, it's a lot like a computer program or a language. The order of A's, T's, G's, and C's forms a code. This genetic code is read in 3-letter words called codons. Each codon specifies a particular amino acid, which are the building blocks of proteins. A long string of these bases, forming a specific instruction, is what we call a gene. For example, one gene might have the code that tells your body how to make hemoglobin, the protein that carries oxygen in your blood. Another gene might hold the instructions for creating the melanin that determines your skin and hair color. The human genome, which is the complete set of our DNA, contains about 3 billion of these base pairs. If you were to print out the entire sequence in books, it would fill a library of about 200 telephone directory-sized volumes. All of that information is packed into the nucleus of a cell so small you can't even see it. It's an absolutely mind-boggling feat of data storage. This code is what tells your cells what to do, how to grow, and how to function every single second of your life. Now, having the instructions is one thing, but how does the cell actually use them? It's a 2-step process. The first step is called transcription. Since the DNA is super important, it's kept safe inside the cell's nucleus. To get the instructions out to the main part of the cell where the work gets done, a special copy is made. This copy is not DNA. It's a slightly different molecule called RNA, or ribonucleic acid. An enzyme travels along the DNA, reading the code and creating a messenger RNA, or mRNA, strand. Think of it like taking a photo of a single recipe from a giant, precious cookbook that can't leave the library. This mRNA molecule then leaves the nucleus and travels out into the cytoplasm. This is where the second step, called translation, happens. Here, cellular machines called ribosomes read the mRNA sequence, 3 letters at a time. For each 3-letter codon, the ribosome grabs the corresponding amino acid and links them together in a chain. As the ribosome moves down the mRNA strand, the chain of amino acids gets longer and longer. Once it's finished, this chain folds up into a specific, complex 3-dimensional shape, and voila, you have a protein. These proteins are the real workhorses of the cell. They do almost everything. They act as enzymes to speed up chemical reactions, provide structural support like collagen in your skin, transport molecules like hemoglobin, and send signals like hormones. Essentially, DNA holds the blueprints, but proteins are the builders, the mechanics, and the messengers that bring those blueprints to life. This entire process, from a sequence of letters in your DNA to a functional protein doing a job, is happening constantly in trillions of your cells. It's the fundamental process of life, often called the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. But what happens when a cell needs to divide? How does each new cell get its own complete copy of the instruction manual? This is where another incredible process comes in: DNA replication. It's essential for growth, repair, and reproduction. Before a cell divides, it has to make a perfect copy of its entire DNA. The process starts with the DNA double helix unzipping right down the middle, like a zipper. An enzyme breaks the bonds holding the base pairs together, separating the two strands. Now you have two single strands, each with a sequence of bases exposed. Each of these old strands then serves as a template for building a new, complementary strand. Free-floating bases within the nucleus find their matching partners on each template strand. Remember the rule: A pairs with T, and G pairs with C, so wherever there's a G on the old strand, a C will lock into place. Wherever there's an A T will join. Another enzyme, called DNA polymerase, moves along each strand, linking these new bases together and proofreading to make sure there are no mistakes. The end result is two identical DNA double helices, where there was only one before. Each new molecule consists of one of the original strands and one brand new strand. This is called semi-conservative replication, and it is an incredibly efficient and accurate way to copy billions of letters of code with very few errors. Thanks to this process, every time one of your cells divides, the two new daughter cells each receive a complete and perfect set of genetic instructions. So, let's bring it all together. DNA is the blueprint of life, a twisted ladder made of four bases of T, G, and C. The sequence of these bases forms genes, which contain the instructions to build proteins. Through transcription and translation, these instructions are read and used to create the proteins that do all the work in our cells. And through replication, this vital information is copied and passed on, ensuring that life can continue and grow. This amazing molecule is what makes you unique. While we all share about 99.9% of our DNA with every other human, that tiny 0.1% difference is responsible for all the diversity we see in our species. It's what gives you your unique talents, your appearance, and your predispositions. Understanding DNA has also revolutionized our world. In medicine, it allows us to diagnose genetic diseases, develop targeted therapies, and engineer life-saving drugs. In forensics, DNA fingerprinting has become an invaluable tool for solving crimes and identifying individuals. It's even helping us understand our evolutionary history and our connection to all other life on Earth. From the simplest bacterium to the most complex human being, DNA is the thread that connects us all. It has a code written billions of years ago that continues to write the story of life every single day. Thank you so much for joining me on this journey into the world of DNA. I hope you found it as fascinating as I do. If you enjoyed this video and learned something new, please give it a thumbs up and consider subscribing for more deep dives into the wonders of science. Let me know in the comments what molecule you'd like to explore next. Thanks for watching, and see you in the next one.